Okay, so the first thing I want to do today is to hand back the exams. If you got my email last night, not so good. Some people did really well. A lot of people didn't. Let me just get the grade distribution up here in a second. Okay. So here's the grade distribution. The average for the test was 64.9, which isn't really good. I'd like to get that up around like the mid 70s would be perfect, but low to mid 70s, like a C should be average. Grade distribution was a little strange, disappointing. So there was 4 A's, 4 B's, 9 C's, 9 D's, and then 6 D-NEFs. So that's not ideal. The good news is that no matter how bad you did on the first exam, there's still hope. No matter how badly anybody did, you can still pass the course. But you need to do something, like now. My suggestion for you would be, first of all, to try to get a tutor. There's tutors available for free through the Student Success Center. You just go over to the library, and you can't miss it. Student Success Center is there. Tutors are totally free, it won't cost you a cent. Also, another thing you can do, well, you have to be eligible for this, is to go to Student Support Services. There is some paperwork you have to fill out to make sure you're available, but we just got an email from them that they have tutors available as well. So a tutor would definitely be very helpful. Your other options, of course, office hours have been difficult this semester with the restrictions on COVID and everything. But if you want to make an appointment to see me, like a recurring appointment for office hours, you can do that too. Okay, I'll be more than happy to help you. A third thing is, don't neglect the fact that you guys have each other. It might be helpful to set up a study group so that you guys can, get together and study together. But no matter what you choose, whether you get a tutor through Student Support Services, through the Student Success Center, or office hours with me, whatever you do, you need to do something. Students who do something early tend to succeed. Students who just say, like, well, I'll wait. Maybe if I don't do better on the next test, then I'll do something. You're just digging yourself a deeper hole if you do that. So no matter how badly you did, if you do something about it now, you still have a chance to pass. All right? So please, I'm asking you, if you didn't do well and you want to put the effort in to pass, there's resources available to you. Take advantage of them, and you can still salvage the semester. All right, so before I hand these back, let me show you just what I did. So I told you I use test grading software on these. So not the last, so the last page is your answer sheet. The page before that is the student grade report generated by the test grading software. Okay, now this only graded the test part, only the multiple choice questions. So I had to add in the bonus question. So the number circled in red is the bonus, your bonus points plus the points from the multiple choice questions. Okay. Also, if you look on the student grade report, it shows you what you answered and what the correct answer was. So because you have this available to you, I'm not going to go over all the questions in class. You can just go over this at your own leisure. But do check it over. I mean, the computer isn't perfect. Sometimes it'll record something as wrong when in fact it was right. My math isn't perfect, so double check my math, make sure I added everything up correctly. And other than that, you want to hang on to this. The reason for that is, remember that we have a comprehensive final in this course. Because we have a comprehensive final, your old exams are going to be your best study resource for the final exam. So hang on to these. When we get to the final time, you want to study your old tests. Definitely want to study your old quizzes as well. So this is something that you need to hang on to. Okay. So I'll tell you what, why don't I come down here and I'll meet you guys halfway. All right, is Caleb Atkins here today? How about Kelsey Atkins? Bethany Yankee? Elizabeth Bales? Not here today. How about Jacob Bishop? Jacob Bishop? Dylan Blaylock? Maybe Jacob? Dylan Blaylock? Claire Bowers? Byron Collins? Seth Croce? Colton Dorsey, I think he's got a military thing. Paula, let's see her. Caitlin Fitchwell. Chris Gray. Anthony Gilly. Harley Hall. Tia Harris. Alandis Hill. Noah Hill. Deanna Lawrence. Selena Humphrey. Selena, oh, there she is. Savannah Keaton. Savannah. Levi Ledger. Diego Lopez. Elizabeth Flores. Preston Harris. Delaney Leader. Laura Murray. Mackenzie Simmons. Policy. Katie Stevens. Katie Stevens. Bethany Silver. Timothy, pick up the last name. Timothy Trade. Hope Tyrese. Hope. Angela Van Horn. Brooke Ward. Cassidy Wells. Cassidy not here. Nathan Williams. Joseph Wilson. Ashley, is that wrong? I'm sorry, I can't pronounce your first name. All right, is anybody here who did not get their stamp back? Okay, so like I said, double check how the computer read the scans, double check my math, and double check the bonus question. And if you find any problems, let me know, please. Okay, so we've got just a little bit of chapter 5 to finish up. Okay, I see some questions on Zoom. Can I get the test back next time? Yeah, anybody who's attending by Zoom today and didn't get your test back, I'll try to remember to bring them to lecture with me. If not, stop by my office. And if I'm in my office, I'll be happy to get the exam back to you. Okay. So let's get back to chapter 5. There's just a little bit of chapter 5 to finish up, and then we'll be starting chapter 6 today. Okay, so this is where we left off. We were talking about some general characteristics of enzymes. Enzymes are very specific, and they only recognize specific reactants called substrates. So you might recall that terminology. The reactant that's acted on by an enzyme is referred to as that enzyme's substrate. And enzymes are very specific for their substrates. If you look at this particular diagram, so we have the enzyme here in light blue, the substrate in the purplish color. You can see that the shape of the enzyme's active site is very complementary to the shape of the substrate. This is important because the weak intermolecular forces that hold the enzyme, that hold the substrate in the enzyme's active site act over only very short distances. Okay, so they have to fit together very closely in order for these weak molecular interactions to come into play. Okay, and also these weak molecular interactions are going to line up too. So if we have like a proton donor and a proton acceptor that can make a hydrogen bond, they're going to come close enough together to form that bond when the substrate fits into the active site. Okay, and we had some nice animations. Unfortunately, all the animations I use are Flash animations, and Flash is kaput now. But hopefully, if you guys want to play those, I gave you a link to that emulator that can still play Flash animations. And I find those to be very helpful. Just remember, like in that first animation we did, there are some mistakes. That first one, I can't believe that they didn't balance that equation, but they did not balance that equation. So just be aware of that. Okay, electron-carrying cofactors. So you may recall that cofactors are small organic molecules that help enzymes, often by carrying hydrogens, electrons, or functional groups. If they're carrying electrons, we have certain terms for these. So NADP is an electron carrier in photosynthesis. NAD and FAD are electron carriers in aerobic respiration, and we'll talk a whole lot more about all three of these over the next two chapters. Cofactors carrying electrons are said to be reduced. Cofactors without electrons are said to be oxidized. It's pretty easy to tell whether a cofactor is oxidized and reduced, at least in biology. When a cofactor gets electrons, usually one or two hydrogens come along with it. So if it has hydrogens, we know that it's in its reduced state. So carrying electrons is reduced. If they don't have electrons, they're said to be oxidized. Okay, and I have a couple mnemonic devices here to help you. So you might recall when we were doing levels of classification in nature, I gave you guys the mnemonic device, do kings play chess on Fridays, generally speaking, and each word stood for one of the classifications. So in order to remember whether something is oxidized or reduced, we can use these two mnemonics, oil rig. So oil oxidation is losing electrons, reduction is gaining electrons. Okay, so in other words, if it has electrons, it's in a reduced state. If it has no electrons, it's in an oxidized state. Another mnemonic device, if you prefer, Leo the lion goes grr. Leo, losing electrons, is oxidized. Gaining electrons is reduced. All right? So just a little trick to help you remember that. Like I said, in metabolic reactions, electrons usually come along with at least one hydrogen. So you can see the difference between these cofactors in their oxidized and reduced states. So here they are in their oxidized states, okay, not carrying electrons. So we've got NAD plus, NADP plus, FAD. In the reduced state, again, the electrons come along with one or two hydrogens. Reduced, so carrying electrons, it's NADPH. When NAD is reduced, it's NADH, carrying electrons. And when FAD is reduced, it's reduced to FADH2. Okay, finally, again, you might recall that another thing that's important in metabolic reactions are energy carriers. The most important of the energy carriers by far is a molecule called ATP. And like I've told you several times already, and I'll tell you a lot more before the end of the semester, as far as the cell is concerned, ATP is energy. Okay, ATP equals energy. It's the energy currency of the cell. It contains high energy bonds that can be hydrolyzed or broken, and we get energy out. So these high energy bonds exist between the first and second phosphates and the second and third phosphates. Okay, so these are the phosphates over here. So we have 3 phosphate functional groups. This won't be on the test, but usually to keep them straight, we denote these alpha, beta, and gamma. So the alpha phosphate is the one closest to the 5-carbon sugar here. The beta phosphate is the next one out. And the gamma phosphate is the last one. So high energy phosphoanhydride bonds exist between the first and second phosphate, or the alpha and the beta phosphate, and another one between the beta and the gamma phosphate. Okay, so these two bonds here, these are high energy. All right, and specifically, they're called phosphoanhydride bonds, but you guys don't need to know that for this test. Oh, why does it do? Only this computer does this to me. And now there'll be all kinds of scribbles. Yep. Okay. I don't know why this computer does that, but you guys get the idea. Oh, wait a minute. There we go. I can go back to drawing now. High energy bonds between the alpha and the beta and the gamma phosphates. Okay? These bonds can be hydrolyzed in order to do work. And again, some of the examples of the work that can be done with the energy from ATP is driving forward endergonic reactions. So if there's a reaction that needs a net input of energy, we can hydrolyze ATP for that. or active transport. Remember, in active transport, we're moving things across a membrane against their concentration gradient. For that, we need a net input of energy, which can come from ATP. All right, that is the last slide for chapter 5. Does anybody have any questions on anything we covered in chapter 5? Anything you're not sure of? Okay, if there's no questions on chapter 5, we're going to start chapter 6 today. So I like to warn my classes before chapter 6 that this is going to be, I don't know if I should say one of the hardest chapters or the most difficult chapter that we're going to cover the entire semester. Okay, students really seem to struggle with chapter 6 and chapter 7 on test number 2. So I'm going to try to go really slow. You'll probably notice me repeating myself a lot as I go over things. I do that on purpose. After you repeat something enough, sometimes it sticks in your head. So again, expect this material to be kind of challenging, but I will be going slower and I'll be repeating myself a little more to try to get it to stick. Okay, so like the title said, photosynthesis is where it starts. So remember, the flow of energy through the earth, for the most part, comes from the sun, flows through the producers, then to the consumers and decomposers as well. So photosynthesis is where primary producers get their energy. Again, the primary producers use the energy from sunlight to make their own food. This is carried out by plants and some protistans. In the case of algae, you may remember that those huge Sargasso seaweeds I showed you early, those are actually protistans that do photosynthesis. Photosynthesis uses the energy of sunlight to make carbohydrates from carbon dioxide, CO2, and water. Okay? So again, this is a biosynthetic reaction, okay? And endergonic, energy in, right? We're taking small, low energy molecules like CO2 and H2O, and we're using that to make glucose, which is a large, high energy molecule. Okay, glucose is C6H12O6. Oh, damn, I hate this. Stop it, stop it. Sorry about that. Anyway, that was supposed to be C6H12O6 is the chemical formula for glucose. All right. So again, my point being that we're taking small, low energy molecules, using the energy from sunlight to build big, high energy molecules that can be used as a food source. Okay, photosynthesis occurs in organelles called chloroplasts, which we've already talked about a little bit. The structure of chloroplasts, they have 3 membranes, 2 outer membranes, we can see here. So here's the outer membrane and the inner outer membrane. And then the innermost membrane is kind of folded in an unusual way. This is called the thylakoid membrane. And the thylakoid membrane is folded into stacked, interconnected discs. Okay, so 3 membranes, 2 outer membranes, and the thylakoid membrane that's folded into stacked, interconnected discs. These are called thylakoid discs. Okay, and I also want to mention before we go to the next slide, the fluid-filled space between the innermost outer membrane and the pardon me, and the thylakoid membrane. This is called the stroma. Okay, so those are the three things I want you to take away from this. We've got two outer membranes, thylakoid membrane, and the stroma, which is the fluid-filled space between the thylakoid and the innermost of the two outer membranes. Okay, here's another picture of a chloroplast. Up here in the top, that looks like a light micrograph of a elodea leaf. So the weather kind of messed up our shipments last week. I know if you're in my Wednesday section, we didn't get to look at elodea leaf because it didn't come until Thursday. But if you were able to look at elodea leaf, it probably looked a lot like this. So you can see elodea leaf is neat because the leaves are only one cell thick. So when you put them on a microscope slide, you can visualize what's inside the cell really easily. And you can see these green granules, of course, are the chloroplasts. Again, if we look at blow up of the chloroplast, two outer membranes, thylakoid membrane folded into thylakoid discs, and the fluid-filled space called the stroma. Inside of those thylakoid discs is a place called the thylakoid compartment. All right, so let's talk about the reaction itself, photosynthesis. So the overall net reaction is we take 12 water molecules, 12 H2O, plus 6 CO2, and that gives us 1 glucose, C6H12O6, plus 6 O2. So remember, like, you know, the Amazon rainforests are the lungs of the planet because they make oxygen. Obviously, that's a place where a lot of photosynthesis happens. And 6 H2O. So remember that first animation that we did on Monday. This is what they were missing. This is so they omitted this, and that's why the equation wasn't balanced. We can break down photosynthesis into two stages. We have the light-dependent reactions and the light-independent reactions. Both of these stages take place in the chloroplast. Okay, now, this is kind of the new terminology, light-dependent and light-independent reactions. These used to be called the light and the dark reactions, but actually the light-independent reactions can happen if the sun is shining or not. In fact, it happens all the time, whether the sun is shining or not. So now we call them light-dependent and light-independent, which is a little more accurate. Okay, so we can break this reaction down into two stages, light-dependent reactions and light-independent reactions. All right, so let's talk a little bit about sunlight now. So the visible light that we get from the sun is a type of electromagnetic radiation. In fact, it represents just a small sliver of the entire range of different types of electromagnetic radiation. All types of electromagnetic radiation travel in waves. Different types of electromagnetic waves have different wavelengths. Okay, so here where we see the colors of the spectrum, this is the narrow sliver of the electromagnetic radiation that we can actually sense and see with our eyes. Okay, you can see there's a whole lot in the electromagnetic spectrum that we can't see. Electromagnetic radiation with the shortest wavelength are gamma rays. So you can see that gamma rays, we have some wavelengths 10 to the -6 nanometers. That's extremely small. A nanometer is a billionth of a meter. And 10 to the negative sixth of those, that's a millionth of a billionth of a meter. So we're talking teeny, teeny, tiny here. On the other end of the spectrum, we have wavelengths that can be quite large. These are radio waves. So we have wavelengths of 100 meters or even bigger, a kilometer, 10 kilometers, 100 kilometers. Okay, so these are wavelengths that are quite large. Okay, shorter wavelengths correlate to high energy electromagnetic radiation. Long wavelengths correlate to low energy electromagnetic radiation. Okay, so the highest energy of all the electromagnetic radiation are gamma rays. So gamma rays have a very short wavelength, making them very high energy. Radio waves, on the other hand, have a very long wavelength and are very low energy. White light, so white light is right here, that little sliver of electromagnetic radiation that we can actually see. White light is made up of a spectrum of different wavelengths, each corresponding to a different color. Okay, so we can see generally wavelengths from about 400 nanometers to 700 nanometers. And you can see each of these wavelengths. So 400 nanometers corresponds to violet light. 700 nanometers corresponds to red light. And we have different wavelengths in between. You may recall from lab that green light absorbs at 525 nanometers. So each of these different colors corresponds to a different wavelength. Okay, pigments. A pigment is a molecule that absorbs light. Different pigments absorb different wavelengths of light. Chlorophylls are the primary photosynthetic pigments. So again, if you think back to lab, where we measured the amount of red food dye. So red food dye is a pigment that absorbs at 525 nanometers in the green wavelength. Chlorophylls are the primary photosynthetic pigments. And just like with the red food dye, they have certain wavelengths where they absorb light. Chlorophylls absorb mainly blue, indigo, violet, red, and orange wavelengths. So this double peak here, these are the wavelengths that chlorophyll absorbs light. Okay, so they absorb in blue, indigo, violet, which is this peak right here, and then red and orange, which is this peak over here. Yellow and green, which is kind of in this valley between the two peaks, are not absorbed well by chlorophylls. So chlorophylls transmit green and yellow rather than absorbing it. So again, if you think back to when we were measuring the red food dye transmits red light, so it appears red. Chlorophylls transmit yellow and green light, so they appear green. Right? So this is what makes plants appear green. It's because chlorophylls transmit in the yellow and green wavelengths. Okay, so yellow and green wavelengths are not absorbed well by chlorophylls. These colors are transmitted, and that's why plants are green. Okay, so next, we're going to talk about, remember that there's two stages to photosynthesis. We have the light-dependent reactions and the light-independent reactions. So next, we're going to talk about what goes on in the light-dependent reactions. But first, I want to talk a little bit about photosystems. So there are two photosystems that make up the light-dependent reactions. We have photosystem 1 and photosystem 2. So if we look over here, first comes photosystem 2, and next comes photosystem 1. So they're backwards, they're reversed. So you might think, well, why, that doesn't make any sense. Why does photosystem 1 come first and photosystem 2 come second? Well, they were named after the order they were discovered. So photosystem 2 was discovered first. I'm sorry, photosystem 1 was discovered first. And then after photosystem 2 was discovered, they figured out that actually comes second in the order of reactions in the light-dependent reactions. Okay, so we have two photosystems, photosystem 2 and photosystem 1. I'm going to switch over to the whiteboard for a little bit. Okay, so let's talk about the parts of a photosystem. So we have two photosystems, photosystem 2 and photosystem 1. Each photosystem has a light harvesting complex. Okay, sometimes light harvesting complex is abbreviated LHC. Each one has a reaction center. And each one has an electron transport chain. Now, a light harvesting complex, this is an array of chlorophyll molecules. Okay, this is an array of chlorophyll molecules, and it does exactly what its name suggests it does. So it's an array of chlorophyll molecules that harvests the energy from light. So it actually captures light. Second, we have a reaction center. Now, the reaction center is a specialized chlorophyll molecule that we find in the light harvesting complex. And finally, we have an electron transport chain. An electron transport chain is a series of components that pass electrons to each other stepwise. Okay. So electron transport, we have a series of components that pass electrons one to the other. So they just pass it, electron goes from one to the other, to the other, to the other, etc. Okay, so these are the three things that make up a photosystem. Okay, let's switch back to the PowerPoint. Okay, so in this diagram, we can see the three parts of an electron transport chain. So here's photosystem one. This series, why am I not drawing? Okay, so these green dots represent the chlorophyll molecules in the light harvesting complex of photosystem 2. You can see within the light harvesting complex, there's this special, I can't see without my glasses, there's this special molecule called P680. Okay, P680 is the reaction center of photosystem 2. Then these arrows, these represent the electron transport chain of photosystem 2. Okay. Same thing for photosystem 1. We have the light harvesting complex here. Within the light harvesting complex is the reaction center. The reaction center in photosystem 1 is a molecule called P700. And then we have a shorter electron transport chain over here. Okay, so always those three things: light harvesting complex, reaction center, and electron transport chain. Okay, so now let's talk about what happens when in the light-dependent reactions. So first, well, number one doesn't really start at the beginning. So let me start at the beginning. So the first thing that happens is a photon of light is captured by the light harvesting complex of photosystem 2. Let's go back to the whiteboard briefly. Does anybody know what a photon is? Yes.
A light particle.
Exactly, it's a light particle. See if I have them down here to the right. So a photon is a particle of light. Now in physics, I just told you that electromagnetic radiation was waves, right? That's true, and the part that they're a particle is true also. In physics, light is one of those weird things that sometimes it acts like a wave, sometimes it acts like a particle. In this case, talking about a photon of light, this is where it's acting like a particle, okay? So first thing that happens is the light harvesting complex of photosystem 2 captures a photon or a particle of light. Now, the next thing that happens is that photon is going to start bouncing randomly between the chlorophyll molecules in the light harvesting complex. So it's going to bounce around randomly for a while. Eventually, though, it's going to encounter the reaction center. Okay, the reaction center of photosystem 2 is a molecule called P680. Now, when the photon encounters the reaction center, it's going to put 2 electrons in an excited state. So it's going to put these 2 electrons in a higher energy state. And they'll actually be ejected from photosystem, from the reaction center of photosystem 2. So we can see that. So this arrow represents the 2 electrons that have been ejected from P680 because the photon put them in a higher energy state, an excited state. Okay? Then those electrons in the high energy state are going to enter the electron transport chain of photosystem 2. All right. Now I'm not gonna go along with this. There's another thing I want to mention. We need to replace those 2 electrons that were lost from P680. The way that we're going to replace those two lost electrons is by splitting a molecule of water. Okay, let's go back to the whiteboard again. So P680 splits a molecule of water. So we can split a molecule of H2O into 2 protons, 2 electrons, and 1/2 of an O2 molecule. The 2 electrons we can use to replace those that were lost when 2 electrons were ejected from P680 into a higher energy state. Okay? All right. I see there's only 1 minute left. I think that's a pretty good place to stop. So we'll continue with photosynthesis on Friday. Again, remember what I told you. If you didn't do well on test 1, please check out the Student Success Center or Student Support Services, or set something up with me, but please do something. Quiz today. Remember, we're having quizzes Wednesdays and Fridays, so expect the quiz to be online by about noon today, and that's all I have for you. If there's no questions from the people on Zoom, I'm going to stop the meeting now, and I'll see you guys Friday. You too.